Hi, this is Lija here. Today we will discuss gene transfer techniques. Gene transfer involves the introduction of foreign DNA into a host cell. But our cell membrane is a sheet-like assembly of amphipathic molecules that allow only the controlled exchange of materials among the different parts of a cell with its immediate surroundings. Since DNA is an anionic polymer of large molecular weight, hydrophilic and sensitive to nuclear degradation, in biological matrices they cannot easily cross the physical barrier of membrane and enter the cells unless assisted. So techniques are available for the introduction of DNA into many different cell types, which is used very widely both in basic research and applied biology. In research, the transfer of DNA into animal cells is an indispensable tool for gene cloning, the study of gene function and regulation, and the production of small amount of recombinant proteins. Although in many cases the aim of a gene transfer experiment is to express the introduced genetic construct, that is the transgene, in the recipient cells and provide those cells with a gain of function. Gene transfer can also be used to disrupt or inactivate a particular endogenous gene or to perform specific genome modifications. The applications of gene transfer are manifold and range from the use of mammalian and insect cell cultures for the large-scale commercial production of recombinant antibodies and vaccines to the transfer of DNA into human patients for the correction or prevention of diseases, a field called gene medicine or gene therapy. Gene transfer to animal cell is also the first step in the creation of genetically modified whole animals, in which every cell or a specific target population of cells carries a particular alteration. Before considering what cells do with gene delivery complexes, one should consider the makeup of gene delivery vehicles. So we will begin by organizing the gene delivery vehicles and methods into groups. Broadly, we can classify them as physical, chemical and biological methods. Historically, the first gene transfer protocols to be developed for animal cells did not use biological vectors such as bacteria or virus to carry the exogenous nucleic acids across the cell membrane. Instead, naked DNA was mixed with particular chemicals to form synthetic complexes which would either interact with the cell membrane and promote uptake by endocytosis or fuse with the membrane and deliver the DNA directly into the cytoplasm. Since the late 1960s, such chemical transfection methods have been widely used and many different complex forming agents have been described. Various charged chemical compounds can be used to facilitate DNA transfer directly into the cell. These synthetic compounds are introduced near the vicinity of the recipient cells, thereby disturbing the cell membrane, widening the pore size and allowing the passage of DNA into the cell. An ideal chemical used for DNA transfer should have the ability to facilitate transport of DNA across the plasma membrane, and it can protect the DNA against nuclease degradation. The ability of mammalian cells to take up exogenously supplied DNA from their culture medium was first reported by Sibiliska and Sibiliski in 1962. They used total uncloned genomic DNA to transfect human cells deficient for the enzyme HPRT, that is hypoxanthine guanine phosphoribosylase transferase. Rare HPRT positive cells, which had presumably taken up the fragments of DNA containing the functional gene, were identified by selection on HAT medium. At this time, the actual mechanism of DNA uptake was not understood. Much later, it was appreciated that the successful DNA transfer in such experiments was dependent on the formation of a fine DNA calcium phosphate coprecipitate, which first settles on the cells and is then internalized. The precipitate must be formed freshly at the time of transfection. It is thought that small granules of calcium phosphate associated with DNA are taken up by endocytosis and transported to the nucleus, where some DNA escapes and can be expressed. This technique became generally accepted after its application by Graham and Van der Eep to the analysis of infectivity of adenoviral DNA. It is now established as a general method for the introduction of DNA into a wide range of cell types in culture. The principle of the technique is that DNA in a buffered phosphate solution is mixed gently with calcium chloride, which causes the formation of a fine DNA calcium phosphate coprecipitate. The precipitate settles onto the cells and some of the particles are taken up by endocytosis. Optimate factors like amount of DNA in the precipitate, the length of time for precipitation reaction, and exposure of cells to the precipitate all need to be determined for efficient transfection of the cells. It is a simple and inexpensive method which can be used for stable as well as transient transfection. The calcium phosphate method is applicable in many cell types, but some cell lines are adversely affected by the coprecipitate due to its toxicity and are hence difficult to transfect. Moreover, since the precipitate must coat the cells, this method is suitable only for cells growing in monolayers, not those growing in suspension or as clumps. Alternative chemical transfection methods have been developed to address this problem. One such method utilizes DEA dextran, that is diethyl aminoethyl dextran, which is a soluble polycationic carbohydrate that promotes the interaction between DNA and the endocytotic machinery of the cell. In this method, the negatively charged DNA and the positively charged DEA dextran form aggregates through electrostatic interaction. A slight excess of DEA dextran in mixture results in net positive charge in the DEA dextran DNA complex, and these complexes, when added to the cells, bind to the negatively charged plasma membrane and get internalized through endocytosis. This technique was initially devised to introduce viral DNA into cells, but was later adopted as a method for plasmid DNA transfer. The efficiency of the original procedure was improved by Lopata and Sussman et al. By adding after treatments such as osmotic shock or exposure to chloroquine to inhibit the acidification of endosomal vesicles, etc. Like the calcium phosphate method, the reagents are inexpensive and the procedure is simple and efficient. In the best cases, the method can achieve up to 80% transient transfection efficiency. Although efficient for the transient transfection of many cell types, DEA dextran cannot be used to generate stably transformed cell lines. Moreover, the complexes are very small compared to the particles formed during calcium phosphate transfection, and one consequence of this is that much less DNA can be used in such transfection experiments. One drawback of these methods, however, is that they are generally inefficient for gene transfer in vivo. An alternative to chemical transfection procedure is to package DNA inside a phospholipid vesicle which interact with the target cell membrane and facilitate DNA uptake. Liposomes are hydrophobic, unilaminar phospholipid vesicles into which DNA can be packaged to form a fusogenic particle. When mixed with the cells in culture, such vesicles can fuse with the cell membrane and deliver DNA directly into the cytoplasm, a delivery mechanism similar to that of cell and protoplast fusion. Gene transfer mediated by liposomes was first described by Fenchler in 1980, and the original liposome transfection technique were no more efficient than calcium phosphate transfection and suffered the further disadvantage that the preparation of DNA containing liposomes was complicated and labor intensive. One particular advantage of this method, however, was the ability to transform the mouse cells in vivo by injecting liposomes into the tail vein. Indeed, liposome mediated transfection was the first non-viral technique devised specifically for in vivo DNA transfer, making it suitable for gene therapy. The efficiency of liposome mediated transfection can be enhanced by incorporating viral proteins that facilitate the active fusion between viral envelope and cell membranes. Such fusogenic particles have been termed virosomes. A breakthrough in lipid mediated transfection came with the development of cationic or neutral lipid messengers, which spontaneously associate with negatively charged DNA to form complexes known as lipoplexes. The first cationic lipid compound to be described was DOTMA, which forms liposomes when mixed with water but assembles into spontaneous lipoplexes with DNA. Residual positive charge in the complex then interact with negatively charged molecules on the cell surface, facilitating uptake of exogenous DNA by endocytosis. Technically, this is one of the simplest transfection protocols. Lipid DNA complexes are prepared simply by mixing DNA with the lipid preparation in serum-free medium. Then the mixture is added to the cells. This facilitates rapid and efficient DNA uptake and is gentle enough to be applicable to both conventional plasmid vectors and large constructs such as bacteria and yeast artificial chromosomes. Liposuction, as this technique has become known, is highly reproducible and extremely efficient for both transient and stable transfection. It allows up to 90% of cells in culture to be transiently transfected and demonstrate stable transfection efficiencies up to 20-fold greater than standard chemical transfection methods. One drawback to this approach is that the lipids are usually difficult to prepare in the laboratory. They must therefore be purchased from a commercial source and they are very expensive. There are now many different lipid preparations available from different suppliers varying in price, efficiency and suitability for different cell lines. This is all about the chemical methods of gene transfer techniques. We will discuss the physical and biological methods in yet another video. Thank you for watching this video. Please share and subscribe.